Hi, my name is Sepideh Sadegh. I am a graduate student at Technical University of Munich. I'm going to shortly present our paper entitled Exploring the SARS-CoV-2 virus host drug interactome for drug repurposing, which is recently accepted in Nature Communications and already in press. First, a short simplified introduction to the SARS virus life cycle in the human cells. For the virus to cause the disease, it first needs to enter the cells through receptors on the surface of the cells. In the host cells, the virus releases its RNA, which contains all the information needed to replicate itself. Host proteins create viral proteins and position them to assemble new viruses. Once genetic material is replicated, the virus can be released again and start infecting new cells. To stop the virus from spreading, drugs need to inhibit the replication of the virus. The common approach is to target viral proteins directly and restrict the assembly of the new viruses. These drugs have to be developed from scratch for each virus. This is associated with huge costs and time-consuming research and approval processes. A fast alternative is the repurposing of the already existing and approved drugs for applications in other diseases. Since the target proteins of these drugs are already known, this information can be connected to the virus-host interactions of new diseases. This facilitates the search for drugs, as there might already be drugs on the market to inhibit important direct interactions of the virus and host cells, or even whole pathways that play a role in the progression of the disease. Identifying such potential pathways or interaction is part of the research area called network and systems medicine. So how are we utilizing systems medicine information and graph algorithms to find such potential pathways and subsequently repurposable drugs? To this end, we developed Coronavirus Explorer, or COVEX. COVEX allows visual exploration of the virus's interactome and implements system medicine algorithm for network-based prediction of drug candidates. In COVEX web application, we use experimentally found interactions between the SARS virus and human host proteins in combination with non-protein-protein interactions that occur in the human cells and protein targets of already known drugs. The analysis always starts with a set of proteins which have to be selected by the user. Here, expert knowledge about virus replication, immune-related biological processes, or drug mechanisms can be applied to compile a set of host or viral proteins, referred to as seeds. This set can include some or all of the viral proteins and/or direct host interactors. Alternatively, the user can select proteins with high expression in a tissue of interest, for example, lung, or provide a custom list of seed proteins. This latter option is especially interesting because the user-provided list of seed proteins could contain host proteins involved in a symptom-related pathways or host proteins which are differentially expressed in COVID-19 patients. After selecting the seeds, the user can choose among 6 network algorithms to individuate potential drug targets, which is host proteins involved in virus host pathways which are relevant to the selected seeds. While these algorithms differ with regarding to their mathematical underpinnings, they all aim at extracting pathways that connect the selected seeds. The returned host proteins can then be annotated with or filtered by tissue-specific expression values. Moreover, gProfiler can be called to run enrichment analysis on the found drug targets. The last step is to individuate drugs targeting the discovered drug targets from step 2. For this, COVEX provides these 4 different network algorithms. All of these algorithms extract ranked lists of drugs that hit directly or in the close vicinity of the found drug targets and are hence candidates for drug repurposing. In our paper, you can find more details on the application scenarios, starting with 4 hypotheses and ending with different drug repurposing candidates, as well as a short discussion on how to prioritize them. Here you can see an overview of the computational methods used in COVEX. We implemented a modified version of the standard centralities. Here we compute the centralities with regarding to only the selected set of seed nodes and not all the nodes in the network. TrustRank ranks nodes in a network based on how well they are connected to a trusted set of seed nodes. It is a variant of Google's PageRank algorithm, where trust is iteratively propagated from seed nodes to neighboring nodes using the network structure. Key pathway miner is a network enrichment tool that identifies condition-specific subnetworks or key pathways. In COVAX, we utilize the key pathway miner web service to extract a maximally connected subnetwork starting from seeds. One of the COVAX algorithms specifically tailored to the network medicine context is multi-Steiner trees. The Steiner tree problem is a combinatorial optimization problem. It aims at finding a subgraph of minimum cost connecting a given set of seed nodes. However, this solution is usually non-unique. For COVAX, we have developed a weighted multi-Steiner trees method which aggregates several non-unique approximates of Steiner trees into a subgraph. So to give you a summary, since the development of safe and effective vaccines is a time-consuming process, the alternative to mitigate the damage by the SARS-CoV-2 pandemic is to quickly identify agents for the treatment and control of COVID-19 symptoms. A particular interest lies in drug repurposing, since already approved drugs can go through shortened clinical trials within months rather than years. Systems and network medicine offer alternative approaches where the process of drug target discovery is driven by computational data mining methods utilizing molecular interaction networks. This data-driven process can produce a list of promising drug candidates targeting host proteins in close proximity and mechanistically related to virus interacting proteins. With COVAX, we seek to make this network medicine approach widely available to the community. You can find our platform in the following link. Thank you.